Continue with the basic radicals analysis under the chemistry experiment salt analysis topic for class 11th. We have already discussed till group 1. I have already shared in the channel till group 1. Now this is the second group cation that I am discussing. There are two cations here, copper and arsenic, but arsenic is removed from the syllabus. So I am discussing only copper here. This will be experiment number 14. So the aim of the experiment is to test the presence of copper ion, that is Cu2 plus, we call it cupric ion, in the given salt. First, the physical tests, as you have to do it when you are actually going to do it in the lab. Mostly the color of the copper salts is bluish or greenish. There are very rare salts like copper iodide is one, which is having off-white kind of color that will be formed here in one of the reactions as a precipitate. But generally the salts that we use, they are colored. You have some knowledge of the coordination compounds, and there is a full chapter on it in class 12. So then you will understand that why these salts are colored. Like copper sulfate in the anhydrous form is white, but if you take hydrated copper sulfate, it has a particular blue color. So that is because of BB transition. So you will be studying about it in detail in class 12. Right now, I'm just discussing this under salt analysis, that the color, smell, particular smell, solubility, these are the physical tests that you have to do for every salt when you start it. Because if you know the characteristics of color, smell, and solubility, then you have some idea that in which direction you have to work. So here, the first test, chemical test would be, you will be taking the aqueous solution of the salt and add dilute HCl to it. Dilute HCl to make the medium acidic, and then you will pass hydrogen sulfide gas through this. If you make use of dilute HCl and H2S, this combination is used as group reagent for group 2. That means to identify whether the group 2 cations are present in the given salt or not. This is the first test that we carry out. So H2S in acidic medium is the reagent, group reagent for second group. If it is copper, you will get a black precipitate of copper sulfide, CuS. So we'll write it may be Cu2 plus. Next test, which will confirm the presence of Cu2 plus, would be, we'll collect the black precipitate in a test tube and dissolve it in 50% nitric acid. And then you divide the solution into 3 parts. To the first part, we'll add ammonium hydroxide solution, and it will give you a blue coloration, blue solution in the test tube. So initially also, the copper salt that we are using in the lab is blue in color, so it will give you the blue color finally. So we'll write it may be Cu2 plus. Still we have to carry out tests to confirm it. To the second part, we'll add potassium iodide solution. It will give you white, or you may write off-white precipitate of copper iodide. That means it is confirming the presence of copper in it. And to the third part, you will add acetic acid and potassium ferrocyanide. Generally, there is another reagent, potassium ferricyanide, K3FeCN6 in the lab. So be very careful, otherwise you will not get the required color in the test tube. It has to be K4FeCN6, potassium ferrocyanide, and you will get a chocolate brown precipitation, which will confirm again the presence of Cu2 plus in the given salt. And then you'll write the result according to the aim, that the given salt contains copper ion in the bracket symbol Cu2 plus in it. Now the reactions involved. First reaction that we carried out, when we take aqueous solution of copper salt and pass H2S through it in the acidic medium, that is in the presence of dilute HCl, we were getting a black precipitation. So that black precipitation is because of the formation of copper sulfide. These things are very important from Viva point of view, and they will make the concept also very clear that why a particular precipitation is seen, and then when it dissolves, then what happens, how do you get the different color. So you must always write, as I always suggest, the equations just opposite to the test that you have written on the ruled sheet. Just opposite to this on the other sheet, the unruled one, you should write the reaction so that when you are revising it, when you are reading from your file, you can prepare for Viva also side by side and understanding would be much better. Then we dissolve that black precipitation in nitric acid and we got this copper nitrate aqueous solution. Now further, this copper aqueous solution, it was mixed with ammonium hydroxide and it was giving a blue color. That blue solution was because of the formation of tetraamine copper 2 sulfate. If you start with copper sulfate, then this would be the name. Otherwise, you can simply write for copper ion, as we are writing here in the equation, we are not showing any sulfate here. So it would be better to give it the name tetraamine copper ion. We generally use copper sulfate in the lab. So that's why in the equation, if you remove this 2 plus and write SO4 here, the name becomes tetraamine copper. 2 is the oxidation state of copper and sulfate, but in general, you can simply write tetraamine copper 2 ion. Okay. Then the next test was that aqueous solution which you got in the second step, that was divided into 3 parts. So this is what we did with the first part. Now the second part, Ki is added to it, potassium iodide solution, and it will give you an off-white precipitation of copper iodide. This is the confirmatory test, and another confirmatory test is third part of the solution. It was treated with potassium ferrocyanide, this complex, and it will give you another complex, that is copper ferrocyanide, which has a chocolate brown color, hence you get the chocolate brown precipitation. So this completes the experiment related to detection of or identification of Cu2 plus ion in the given salt. Now the next experiment is experiment number 15. This is from group 3 of the cations. Group 3 contains two cations, Al3 plus and Fe3 plus. Now the first will be the physical test as usual. You'll see the color, smell and solubility and write the observation and inference as discussed earlier. Then the chemical test would be, to the aqueous solution of the salt, you will add solid ammonium chloride followed by ammonium hydroxide in excess. Now, this combination of solid NH4Cl and NH4OH, it basically acts as a buffer. It maintains the pH of the solution that you have read in chemical equilibrium chapter, because it is the mixture of a weak base and its salt. So to carry out specific reactions to get a particular precipitation, we need fixed range of pH. So this is helping in that only, because NH4Cl and NH4OH, they have NH4 plus as common ion. So this is how the quantity of OH minus is controlled, which helps in controlling the pH. And when you do this, you will get a gelatinous white precipitate. Gelatinous white precipitate, a light precipitate, which will be floating. It will not settle down in the test tube. And if you do it once, you will never forget. Whenever you get Al and you get this type of precipitation, you can be almost sure that you have got Al3 plus. But yes, definitely you have to carry out the confirmatory test after this. So we will write maybe Al3 plus. Then the next step, once you get that gelatinous white precipitate, is a test which is named as lake test. You will collect the white precipitate and dissolve it in dilute HCl again. Then you add a few drops of blue litmus solution. When you are dissolving the precipitate in dilute HCl, you have made the medium acidic. So what's going to happen when you add a few drops of blue litmus to acidic solution? It is going to turn red. And then add ammonium hydroxide, the base, dropwise till you get the blue color. Once that happens, you will get a blue precipitate, aluminum hydroxide, and it's not originally blue. It will take time to turn blue. Reason being, the aluminum hydroxide that you are getting here is white precipitate and it will be absorbing the blue color of the litmus on it and it will give you the appearance of a lake. That is why it is called lake test, and this test confirms that there is Al3 plus in the given salt. Then we'll write the result. The given salt contains Al3 plus ion in it. The problem which happens here in the test is generally students take large quantity of the precipitate and then they dissolve it in a lot of quantity of HCl. Too much acid is used in, wasted in the process. And then when you add blue litmus, it will definitely turn red. But this particular step will take a lot of time, and generally students say that the test is not coming. We are not able to see the blue coloration. It remains red because you have already added so much of acid to it that it will take a lot of quantity of NH4OH to get neutralized and then make it basic so that the blue color is there. So the most important thing is you should always start every test because you are just testing it. You're not going to measure the quantity or weight that how much I have made, that doesn't matter. So make use of very less quantity of every chemical right from the beginning so that you get the test very quickly and you are not wasting your time in chemicals also. The most important takeaway from here. These are the reactions involved. First, Al3 plus aqueous solution we started with, and we added NH4OH in the presence of NH4Cl, and you'll be getting a gelatinous white precipitate. Once you dissolve that gelatinous white precipitate in HCl, you'll get AlCl3. And AlCl3 again in the lake test, when you are adding NH4OH dropwise to it, again it is turning to AlOH3, which is white precipitate, but it will be absorbing the blue coloration of blue litmus on it. That's why it is going to appear blue later when you complete the test. So that completes the experiment related to identification of Al3 plus in the given salt. This is experiment number 16. To test the presence of ferric ion Fe3 plus in the given salt, it also belongs to third group. So the first physical test will be carried out and you will notice that the color of the solution, the salt that is being given to you, that is generally having reddish brown kind of color. So you can write here and you can identify that this must be iron salt. Generally, you will get white salts, but yes, like I told you in copper case also, you should know the chemistry that will help you in theory as well as practicals. So here, after the physical tests, you will carry out the chemical test. The first test is you will make the aqueous solution of the given salt and add solid NH4Cl and NH4OH solution to it in excess. This is the group reagent NH4Cl and NH4OH for third group. So for Al3 plus also, for Fe3 plus also, on adding the same group reagent because they belong to same group, you will get a precipitation. In case of Al3 plus, it was white. Here it is reddish brown precipitate because of the formation of ferric hydroxide, and it will give you the indication that there may be Fe3 plus in the salt. Now the confirmatory tests, same way. as we have done it for Al3 plus. Here also we'll collect the precipitate and dissolve it in dilute HCl. Then we'll divide it into two parts. This is the first part and this is the second part. The first part, you will add potassium ferrocyanide solution and it will give you an instant deep blue coloration in the test tube or precipitation. To the second part, you will add potassium or ammonium sulfocyanide and it will give you a blood red coloration. Now these tests are so fast, you'll get instant coloration in the test tube and you can say that Fe3 plus is confirmed in both the cases. And generally when you prepare for competitive exams, they ask you such questions that which of the following is not considered when we talk about chemical kinetics, or which of the following reactions are beyond the scope of chemical kinetics. So such reactions which give you instant results, there you cannot measure the rate of the reaction, the speed of the reaction. It's not practically possible to do that. So this is the reason that we say that these do not come under the scope of chemical kinetics. And the result would be the given salt contains ferric ion Fe3 plus in it. Now the chemical reactions involved. First you have taken the Fe3 plus salt, aqueous solution, and added NH4Cl followed by NH4OH in excess, and it will give you a reddish brown precipitation of ferric hydroxide. Then ferric hydroxide is separated, the precipitate, and it is dissolved in HCl. It will give you ferric chloride aqueous. This ferric chloride is then divided into two parts and you add potassium ferrocyanide to one. It will give you ferrocyanide. It has a beautiful blue color. It confirms the presence of Fe3 plus in the given salt. And to the second part, you have to add potassium or ammonium thiocyanate, or we call it sulfocyanide. It will give you the blood red coloration instantly. So these are the two confirmatory tests for Fe3 plus. This completes group 3 also. Now in my next presentation, I'll be talking about the 4 cations present in group 4. Thank you.